Michael here? Ah, for the last speaker, okay. Because I didn't see you earlier and I wasn't sure where you were. Okay. So we're gonna change gears and go away from publishing. I'm sorry we went a little bit over on that. We started just a little bit late. But now we're going to go to overcoming the translational gap in nanomedicine with Theresa Mary Allen, and then into IT after this. And I think this is an interesting segue, even though they don't seem to go together.
Yeah, and then just to finish up on those final points, that's not even to mention the fact that an astonishingly high percentage of work that's published in the medical literature has turned out to be non-reproducible. And that's a whole other kettle of fish that we haven't touched upon. But it is, in my position, in my role and position at the Center of Drug Research and Development, this is a source of frustration when we're trying to translate, because we don't know whether what's being brought to us is even reproducible. And so we have to reproduce it, and drug companies have to reproduce all of this material, again, from scratch, to make sure that they're dealing with a real phenomenon and a real target. So now to actually change and go a little bit from that last remark, which is almost like a preface remark to how do we overcome the translation gap. I want to just do a bit of an introduction, and then most of my talk is going to be actually about a reality check and suggestions towards the end of the talk. So I come from the liposomal field, and it's a field in which there have been a number of products that have been brought to market starting in the 1990s. So this is now quite a number of years ago. So it's really become a very mature field to the extent that we're now having generic products appearing on the market. This is the kind of territory that we've been dealing with, and you're probably familiar with all of these different types of liposomal products that are on the market or approaching the market. So just a brief review to set the stage. Most of the types of drugs are in liposomes or lipid complexes that have come to market with the nanomedicines. And you see that they're mostly anti-cancer drugs. There are 3 versions of the same antifungal drug: an ocular drug, a local anesthetic, an analgesic, which has now ceased marketing, an antibiotic in clinical trials, and some ligand-targeted liposomes or lipidic nanoparticles in clinical trials, and 3 siRNA, at least, products in clinical trials. And the main properties are that they're almost entirely administered IV, and one is for soft tissue. They're classical liposomes, meaning that they're not pegylated, a couple of pegylated ones, one of which is a hydrophobic drug, all of which are monotherapies, meaning that there's only one drug in the nanoparticle. However, there are 2 combination drug particles that are in clinical trials, one triggered release particle, thermodox, and 3 or possibly more now ligand targeted. So that's the spectrum of nanoparticles as far as the market goes at the present time. So and one of the biggest selling of these is Doxil, which where I have been throughout my career involved with the development of Doxil and targeted Doxil. So what is the present situation that we're in? The drug discovery business is really under pressure right now because, first of all, the low-hanging fruit has already been picked. The costs of drug development are increasing enormously. The numbers yet despite this more and more money being spent, the numbers of innovative breakthrough drugs are declining. The costs of the new drugs when they come to the market are escalating, which you'll see some examples of, and the price to benefit ratio, however, is declining. You're spending a lot more money for the drugs, but the benefit that you're getting from them as a patient is actually going down. Drug resistance is increasing and developing more quickly, so the drugs have a shorter period of time that they're going to be giving you a benefit. Generic drugs, because of the development of a lot of these expensive new products, that seems to contribute to the fact that generic drugs are in short supply, and there's not a week or two goes by when I don't hear something in the media that says X drug or Y drug is now no longer is unavailable or back ordered or in short supply. And although we're getting lots of new targets from genomics, proteomics, metabolomics, microbiomics, the translation of this information to developing new drugs basically will lead to development, manufacturing, and regulatory bottlenecks, because we've got a lot of information coming in, but the bottleneck now is going to be how to get that information translated into meaningful new products. So how is this going to affect nanomedicines? Well, future formulations, according to the talks I hear and according to the papers that are published, will include combinations of therapeutics, not only one therapeutic, one or more molecules for site-specific targeting. They will include also biomarker and imaging capabilities, often built into the same particle, and also sensing molecules that can respond to external or internal triggers to control the rate of drug release. And so this means increasing complexity of the nanomedicines that we're predicting will be developed in the future. But as the complexities of the formulations increase, so do the expenses and difficulties associated with their manufacture and their quality control. And also, if you have a complex formulation, then it may be that you don't control all of the intellectual property yourself. So the establishing control of the intellectual property associated with these complex formulations will also increase the cost. So therefore, the development costs for these complex multi-component systems such as nanomedicines are going to be much higher than those that are traditionally seen for the small molecule therapeutics or for the classical liposomes. So we're in a new paradigm right now as far as the drugs business goes. So, and the payers now are asking, the people who pay for these new drugs are asking not only the traditional questions, which were, is this drug safe? And is this drug efficacious? But the payers are now starting to ask, is this drug's level of efficacy worth its price? And so I want to give you a few examples of products where these questions are now triggering a lot of discussion. So Salvati, the new hepatitis C drug that Gilead brought on the market recently, it's a very good drug for hepatitis C. There's no doubt about that. It's causing cures in patients who had been deemed uncurable. But it's expensive. It's $1,000 a pill, $84,000 for a 12-week treatment course. So yes, it's effective, but part of the problem is that hepatitis C is a common disease. There are 4 million patients in the US that could benefit from this treatment. And the cost of this to treat these patients would be $200 to $300 billion. And yet, the amount in the US now that's being spent for all drugs is only $260 billion. So do the math. So there's a huge return on investment for Gilead. Forbes recently estimated that the return on investment for Salvati, who paid $1 billion to acquire this drug, was going to be something like 2,500-fold ROI. And through 2016, they project that there's going to be an 1,800% increase in the cost of treating hepatitis C. Another effective new drug is bedaquiline, effective for resistant tuberculosis, a very good drug, no argument about that, but it costs $30,000 US for one course of treatment. How do we take this to Africa or Asia, where TB is endemic, and actually expect to pay for the patients to be able to get access to this kind of treatment? Now, here's where we're getting into stranger or more difficult territory. We also have a lot of new drugs coming out of antibodies and biologics where the cost of treatment is very expensive. Yervoy, $30,000 per injection, $120,000 per course, adding an increased survival of 3.7 months and an overall survival rate of increased overall response rate of 11%. So the average cost per month per drug of added survival is in the $30,000 to $40,000 range. And so regulatory authorities like NICE in the UK declined to recommend the drug on the grounds that its longer-term benefits were unclear. And then we have the case of Avastin, a different case where it was sold at $60 a dose for cancer, but they reformulated it as Lucentis, and all of a sudden they're selling it for $2,300 a dose for basically the same drug, but for the indication of wet macular degeneration. And then a final example, Solaris for an orphan disease, which is costing about $570,000 a year for treatment. So the payer, NICE again, actually came up with the idea that they wanted to link the market price of the drug to the R&D investment in the drug. And this has important ramifications for the industry and for the patients. So first of all, the kinds of statistics I've just given you have triggered a backlash amongst government, insurance, biotech, and the media against the high cost of these new molecularly targeted and biological products, many of which show only marginal clinical development. But what they're not taking into consideration is the fact that the price that is charged for a new molecule includes the cost of many unsuccessful drugs. So you're not only paying for the drugs that make it to the market and are successful, but you're paying for that pipeline of failures as well. And for the drug companies to stay in business, they need to pay for the failures as well as the successes. So therefore, it's really difficult to separate the costs associated with the development of each specific drug. And therefore, tying the cost of the drug to the R&D for that drug is actually not a workable solution. And also, the profits from some drugs have to justify the increased risk of orphan drug development, where the patient populations are much lower. And we don't want to reward inefficiency in drug development by tying the cost of the drug just to its R&D production. So now to go back maybe to translation of nanomedicines, to keep the cost to benefit ratio within reasonable limits, and this is going to have to apply in our field as well as in other fields. The additional therapeutic benefits that accrue to complex systems need to be substantial. If we're going to make a complex multi-component system, then we need to expect more than marginal benefit from this system. And due to the often poor correlations between therapeutic effects seen in animal models and in clinical outcomes, really, when you look at the results from your animal models, only highly significant increases in efficacy in animal models will be likely to translate to clinical benefits that justify the development costs and the high cost of treatment of these complex multi-component systems. So a lot of these nanomedicines are used in cancer chemotherapy. And in cancer chemotherapy, conventional current cancer chemotherapy almost invariably uses, I can think of very few examples where they're not using combinations of drugs or combinations of drugs and biologics. But currently, all of the approved nanomedicine products are monotherapies, and they're not always going to be competitive with these new combination chemotherapies. And so we're going to have to test them in a situation where they will be competitive. It's going to be hard for them to compete with some of these new combination chemotherapies as a single component monotherapy. And actually, with a lot of the products on the market, decreased toxicity appears to contribute more to the increases in therapeutic index than increased efficacy. And this is particularly obvious in the FDA, where they state that they're looking for increased outcomes. They're not looking for just decreased toxicity. And then here's an example that maybe illustrate what I'm trying to say. We've really made very good progress to date in the treatment of hematological malignancies with conventional combination therapy, things like R-CHOP. So you can get, with R-CHOP, you can get maybe 80%, 90%, 85% cure rates. So even with high cure rates in animal models, even if you hit them out of the park in an animal model, it's going to be hard, long and expensive to power a clinical trial that's going to show significant increases over many of the current combination therapies. So what you have to do is to actually, you're not going to compete against R-CHOP, but what you could possibly do is substitute a liposomal or a nanomedicine drug as one of those components in a current conventional combination chemotherapy, and this is being done quite a lot, but that's going to increase the cost and increase the length of time to do the clinical trials, empower them to actually show that there's going to be an advantage over the significant over the present combination chemotherapy. So given this kind of background that I just presented you with, what are the kinds of things that we can do in our nanomedicine field to have a successful translational outcome? So early on, first of all, we have to look for applications where there's a high unmet medical need and a reasonable population size. And you have to do that before you've made a lot of progress in developing a nanomedicine in order to have an idea of what a potential application might be. Many of us are developing ligand-targeted therapeutics, and we know from a lot of work in animals in particular that they do best where good access to the target cells is possible. So look for applications of ligand-targeted therapeutics in, for example, micrometastatic disease, or in targeting to the vasculature or within the vasculature, or for hematological malignancies, but only those with high unmet medical need, or for targeting nucleic acids where it's mandatory that they get access to the cell, and that can only be helped. They can only get into the cell interior by using an internalizing ligand approach. Keep in mind that most early clinical trials in oncology are done in patients with advanced disease, and this is not necessarily the right niche to prove effectiveness for ligand-targeted therapeutics, because the penetration barrier in these patients with advanced disease and the inability of the nanoparticles to penetrate into the centers of tumors or throughout tumors with advanced disease is not going to lead to a lot of success in the clinic at the, you know, in the phase 1 and phase 2 clinical trials. And so we should be looking for use in adjuvant therapy. For example, ligand-targeted therapeutics seem to be very good at having anti-metastatic effects and in clearing up metastatic disease where the disease is much smaller, where the tumors are much smaller and much more accessible. And so, but however, if you use them in adjuvant therapy, then the clinical trials are much longer and much more expensive. Another thing we can do is try to take advantage of new regulatory authority programs. So the FDA and the EPA have new programs for orphan drugs or for neoadjuvant programs, and those programs can help you actually find a niche where it's going to be easier to get them into the clinic. We also have to work with consultants or experienced nanoparticle manufacturing companies that can help you focus, streamline the product development. And we have one of those in Vancouver that I'm familiar with, Northern Lipids, that a lot of people have been working with to help them get their products to translate. As I approach the end here, consider developing therapeutics where ligand targeting and internalization is essential for activity, like nucleic acids, proteins, and peptides. And consider applying principles of combination therapy at the early stages of development, where you use 2 or more drugs, 2 or more ligands, or are targeting to 2 or more targets. And finally, consider the cost-benefit ratio early in development, because you don't want to be confronted with the fact that you've developed a product and it looks like it's going to be successful, but you just can't afford to develop it and sell it in the end. So the next one, so the last slide is, I still think that many opportunities for new clinical applications exist in the nanomedicine field, but I think it's going to require creativity and rigor to choose the right application and the right clinical niche, so you end up with this kind of a result rather than this result. And this was a photo I took actually last year on an old medieval building in Basel, Switzerland. And I just should finish up by actually thanking the organizers for the opportunity to come here and to have a talk. So thanks, Bea, for inviting me.
Questions?
You know, as you end up with this smile, I'm here. Hi, how are you? Thank you for the talk. Just to be sure that we are ending up with a smile, I just want to emphasize what you said right in the beginning and then through the talk regarding reproducibility and replicability of the publication, which is a major thing, and we don't have really the full answers with that, not theoretically and not technically. And that's again what you mentioned in your talk regarding the to be sure that we are taking the right number of participants in the clinical trials and so on. As we said, we don't have really the right number, therefore we need new understandings, new technologies, new tools that we don't have nowadays mainly. And the last one that I want to remark regarding the translation that you said we are developing a drug, but we don't look on the whole body, or rather the other drugs. We are looking specifically rather than, on the whole. It's not simple, but we have to do that. It should be, as broad as possible approach, and we can do much further than we are doing today.
Thank you. I can't help but agree with you. If you're going to be successful in translating, and the center that I helped to co-found, the Center for Drug Research and Development, that's what we do. We take academic discoveries in the health sciences and we help the academics to translate them to the clinic. And the approach that we take is that we need to know what the, almost from the beginning, what the end application is going to be and what are the implications for development. The more you can know about the process and the more you can get help from clinicians or other senior people in the field who understand the process that from early on, then the more likely you are to be successful. And there, you know, there's a lot of incubators now, and there's a lot of people in universities and in the IP offices and things that have advice that they can give you. Go and talk to them. See them early on. If you think that you've got something that's worth developing and that could be a product, seek help from these individuals and these offices who have, and these centers like my center, who have the knowledge that can impart to you and prevent you from going wrong even at the very early stages. Yes.
Just a, sorry, hi. Thank you for the nice presentation. I would like to go back to this price question of drugs, and especially, let's just take one example which you made, the Yervoy one. So this is over $100,000  4 doses, and you made the point that it is a couple of months survival benefits. So actually this is correct if you take a randomized population.
Yes.
But the big breakthrough of immunotherapy is that there is a 15% response rate in this Yervoy, for example, population, and who survive over 15 years. So this is the efficacy is exceptional, not bad, not okay. Per month, little. It is exceptional for people who can respond. Now, so NICE, the UK regulatory agency, commented when they didn't approve that they would approve if there would be a biomarker or yes. A selection if it's 15% of the patient would be somebody would be able to identify. So what would be the value of a biomarker? My question comes to you. If there would be a diagnostic test which would be able to identify this 15% of the patients, what would, for $100,000 therapy, very successful, very effective therapy, what would be the value of a biomarker?
Absolutely, I think, essential. And as we move forward into personalized medicine, we're going to see that treatment, I mean, it's not going to be any more of this one-size-fits-all, which is the problem with Yervoy. If you've got melanoma, they give it to you, and there's an 85% chance that you won't respond. If you have a biomarker, if you have a diagnostic assay, and you can actually identify the patients that can respond, then there's an economic benefit to that.
So what would be the value of such a biomarker, of such a test, in your view?
Well, I think that as we go forward, we understand now, as we get into the area of personalized medicine, it's going to be essential for all of the drugs that we use and all of the patients that we treat, that we have an understanding from the beginning, based on their genomics or proteomics or something, of whether they're going to be able to respond to the medication that we're giving them, and based on the genomics or proteomics or metabolomics, which medication we should be giving them. Because we've got a situation right now that, you know, if a patient takes Yervoy and they don't respond, then they're spending the money that they could have used to send their kids to college, for example.
Sorry, but perhaps I didn't ask correctly the question. I also understand the value is for the health system is very high, because we can give more effective treatment for lower cost. But if you have a treatment like Yervoy of $100,000, would you be could you justify a diagnostic test for $10,000 or $20,000? This is what was my question.
Yeah, I think the cost of the diagnostic test has to be only a very small percentage of the cost of the treatment. I think that if you can make a diagnostic test and make it fairly cheaply, I think that's going to be ideal. The more expensive the diagnostic test becomes, then I think the less economic value that it will bring to the system. And so I would say that I would expect that the cost of the diagnostics should be really quite low.
So this is interesting because you would save billions of dollars with a diagnostic test, improve healthcare, and you still think that for $100,000 drug, if you can select response, it is not worth $10,000 or $20,000.
Yeah. I'm not a health economist, and so I can't make those kinds of assays, but health economists do make those types of assays, and I think that I don't remember the details. I have read assays where they've determined that if they spend X amount of money on diagnostics, then they can save Y amount of money on the patient population that might otherwise have been treated. And who will not now be treated because it's been determined that they won't respond. Those assays are being done, they have been done, but I just can't give you an example right now off the top of my head.
You're going to be the last one.
Okay, so just for your suggestions about using multiple drugs, multiple ligands. So with the drugs, I would agree partially, because some drugs, you need them to arrive to the site of action in a very timed manner. So you actually don't want them to arrive simultaneously to the site of action. For example, if you are talking about Abraxane and gemcitabine combination, it was proved that actually Abraxane, the addition of Abraxane destroys the stroma, and then you don't need them simultaneously at the site of action. The second comment is the ligand. Actually, by using multiple ligands, you are adding a degree of complexity to the system and a degree of, additional cost. So just if you can comment on that, please.
While he gets set up. Yes, you're absolutely right. I understand that there is an advantage for some drugs to scheduling. I think I would just emphasize the bottom line is that I think that nanomedicines will not be used alone as monotherapy. They will be substituted into combination chemotherapy, and the scheduling will be done according to to what we already know about what could be effective scheduling. And the second point was about multiple ligands. I've been absolutely astonished when they've started doing the genomics of cancer cells at the heterogeneity that's present there. And so it's almost going to be essential to target more than just one cell type expressing one type of antigen if we expect to make progress against this huge heterogeneity, including a number of resistant cells and everything like that. I mean, it boggles the mind when one looks at the data in the literature on this, and you think, how can we possibly make any progress in curing cancer whatsoever in light of this amount of heterogeneity? Well, the fact is we are making progress, and so it's not a barrier that we're not able to overcome. But the more components of this heterogeneous system that we're able to impact, then I think the more likely we are to have a better outcome. And also, of course, that ultimately the cure is a lot to do with the fact that the immune system helps, and it's really, really very promising. The new therapies, like against anti-PD-1 and things like that, that help to deregulate or upregulate T-cell, are making really very good progress in the clinic. So we have to consider that progress on the immune side is going to help this whole situation.
While he's setting up, we'll take one more. Go ahead.
Well, in cancer, you know, the way I've been taught is that the use of placebo in a serious disease like cancer is actually unethical. And so in cancer, the comparator is the best available treatment. So in that trial I mentioned where we have R-CHOP, which is a combination chemotherapy for lymphoma, then if you substitute Oncovin, the liposomal vincristin, into the R-CHOP, the comparator would be R-CHOP. That would be the proper ethical comparator in that particular situation. So in cancer, you always compare, you don't compare to placebo, you compare to the best available therapy that's already known.
But you don't. How are the FDA is always placebo control double-blind study of the new drug?
Well, you control double blind, but one patient goes to the conventional arm, which is the best available treatment, whether it's anthracycline, gemcitabine, or something like that, and then your new drug is in the other arm, and you're double blinded in that situation.
Controlled by placebo.
No, not controlled by placebo.
Not my question. Thank you.
Well, while they're still going, we'll continue this a little bit longer. We're going to run just a little bit long. I'm sorry. Go ahead.
So don't you think that there will be a huge economical benefit for pharmaceutical companies that have failed in phase 3 clinical trials? They've already spent billions, and now to go back to do the biomarker studies to identify the responsive population, and how easy will it be in the future for them to go back to these failed drugs?
Well, usually what seems to be happening right now, and anybody here from Big Pharma can correct me if I'm wrong, but generally as biomarkers are starting to evolve, for example, Theresa with the lung cancer, okay? They had quite a big failure, but when they stratified the patients, they found that there was a certain percentage of patients who responded. And so even at the stage of phase 3, then they were able to start going back and stratify the patients, and then they can apply for approval for now this stratified group of patients, even though in the case of Theresa, you know, that the whole clinical trial would have failed, but they can go back and they can get approval for this stratified patient group. And we're going to see this more and more as personalized medicine evolves, and part of personalized medicine is going to be the ability to identify patients who respond versus everybody gets the same thing.
Hello, Terry. I'm here. I come back to your previous answer and question to the placebo. It's considered to be unethical in cancer evaluation studies to use placebo.
Yes.
Which I can understand, but it's also an obstacle for us to judge the performance of our nanomedicines, because let's say you have a ligand-targeted nanomedicine, you can never show, because you have to compare with the standard medical care, you can never show, for instance, the additional value of a targeting ligand. And this is the type of information we need to know, because you also say it should be an accessible target. Oftentimes maybe targeting ligands are not useful, but in a clinic you cannot evaluate certain elements out of your nanomedicine system because of this placebo requirement, an ethical issue.
You know, okay.
Can you comment on that? Should
Yeah,
we
I don't
try
know.
to implement more of these studies?
Harry, we've been friends a long
studies?
time. What I want to say is that you're not going to like this, but if our nanomedicines can't compete with best available therapy, then they don't belong in the clinic. You know.
It's one issue, of course, that they cannot compete with the best medical care, that they should be better. But are all elements being used in your targeted nanomedicine? Are they justified? Are they doing something? You are adding to the patient maybe elements which are more used, potentially dangerous.
Yes, you're right. You're absolutely right. I mean, there's a clinical trial right now in HER2 Doxil compared to Doxil, okay? That's starting in the clinic. That asks the question is, if you add an antibody HER2 to Doxil, with Doxil already being approved and standard therapy, will you get a better outcome in patients? I think that the answer to your question specifically regarding ligand-targeted liposomes will come out of studies such as those, but they're too early on to know the answer. I can guess that I suspect that they're not going to see enough improved outcome to justify the additional complexity and cost, okay? That's my guess. You can come in 10 years and ask, and if I'm right, you bring me a bottle of champagne, right?